Polymerase chain reaction. It is in vitro amplification of DNA. It was first developed by Carey B. Mullis, and he got the Nobel Prize in the year 1993 for this work. The principle of this technique is based on the fact that DNA replication can be done in vitro using DNA polymerase enzyme. In this technique, we generally use thermostable Taq DNA polymerase because it requires different steps. The three major steps are denaturation, annealing, and amplification. Denaturation occurs at high temperature, and this is performed in an instrument or the machine called as thermal cycler. This works on the Peltier principle. It is a pre-programmed instrument where we can regulate different temperatures for different time intervals, and these can be repeated over different number of cycles. All this can be linked with different temperature in the end and in the beginning. The major component of in vitro amplification, that is polymerase chain reaction. First of all, we require template DNA which is to be amplified or replicated. Then it requires the DNA polymerase which is the key enzyme for DNA replication. In this case, we use thermostable DNA polymerase such as Taq polymerase, which is stable even at high temperature, and it is derived from the Bacterium thermus aquaticus, which is a hot spring bacteria. Now this DNA polymerase requires magnesium or divalent cations for its working. Now the next component which is needed is DNTPs. We require all 4 DNTPs, for example DATP, DCTP, DGTP and DDTP, to be added in the replicated strands. Then to make the condition mimicking the original in vivo condition which are required for Taq polymerase to work, we need optimal pH and concentration of various ions, and these are provided by the Taq buffer or any other polymerase buffer that we are using. Then to start this process of replication as in vivo condition, primers are required. Here also we require the primers. A set of primers are used, forward and reverse primer. These forward and reverse primer is the one which is going to decide the region which is going to be amplified. These are generally 20 to 25 base pair long. एक देखूंगी पीपीटी तभी तो पढ़ूंगी.